Our assignment today is to agree that probably the most important 9 months of our lives are the 9 months before we're born, at least in getting the developmental trajectories started. If that is true, and if I succeed in convincing you of that, if you're not already convinced of that, let me begin by asking how many people know a few important things about that 9-month period of your lives. So how many of you know your birth weight? All right, more or less. By the end, you'll see why I asked that question, because at least in the monkeys that we work with, I can already tell you the age they're going to reach puberty once I know their birth weight, because after all, it's the sum of the 9 months of their first period of growth, and that continues to be fairly steady state in monkeys if we control their rearing conditions. So small babies reach puberty later than ones that are large at birth, as well as you'll see some other consequences of being either small or large. That was the easy question. That was the setup. How many of you know your gestational age at delivery? Oh, that's good. Again, that was just a warm-up question, and you'll see why by the end of this presentation. I asked that because gestational age is one of those programming events, especially if you were a preemie, but as it turns out, even running long has consequences. Babies that run long will be more at risk for allergies and asthma, almost more so than ones that are preemies, even though we always think of preemies. Those were still the easy questions. How many of you know the gestational weight gain of your mother when she was pregnant? You said, thank you. For the younger people in the audience, my mother has passed, so I can no longer ask her these questions, but by the end of this presentation, maybe you'll text your mom and find out. And this was also a relevance for people who study adult participants in their research, because how many of us bother to ask these questions? Maybe we'll ask about childhood trauma or some other events, but rarely do we think to ask, were you a preemie? Were you exposed to dexamethasone? Did your mother have an infection when she was pregnant? You'll see why I'm asking that question as well. And here's the last one. How many of you know whether your mother was happy or stressed during your gestation? Well, I'm impressed. I don't know. And as I said, my mother's passed, so I don't get to ask these questions. But again, you'll see why I asked, because certainly in animal models, stress and other factors like it can influence fetal development. So that really is the sort of the intro to the presentation. As backfill, I'm sure most of you are familiar with this acronym DOHAD, Developmental Origins of Health and Disease. So I'll just touch on that. Most of the work initially focused on prematurity and small for gestational age babies, but you'll see that we're actually going in the other direction. We focus now more on maternal obesity and the large babies. We'll segue then to talk a little bit about immunity and the fact that the immune system and some of the trajectory of immune development begins during the fetal period. There's certainly a thymus from mid-gestation going on. And the point that I'll be trying to convey to you is just as much as the brain is a learning system, the immune system is also one that requires priming and needs to learn, if you will, during the fetal period. And because many of the researchers here are focused on inflammation and infection, I'll show a little bit of our prior research on the effects of flu infections during pregnancy. Transition to the topic of maternal stress, and when we first began to look at that, we were interested in how could maternal stress affect the development of the fetal immune system. How could it affect the brain and emotionality? And then serendipitously, we stumbled upon the fact that one of the important pathways through which these life events and maternal conditions were affecting the fetus was via nutrition, so that it wasn't necessarily always cortisol, the stress hormone that was the mediator, without realizing that we were affecting the transfer of iron from the mother to the fetus with developmental consequences, as you'll see postnatally. And if I don't wear you out and run out of time, I'll talk briefly about the topic that we work on now, which is the gut-brain axis. How does a baby acquire its initial bacteria, which come, as we and others are finding, from the mother, and has that same sort of programming effect on development. But since this is also, as I understand it, aimed for the general audience and the community at large, I just wanted to take a minute to emphasize that there are important societal and policy implications if you agree with this perspective. So I've been working on this topic for a long time, as you can see, going back into the 90s and even before. And I was part of a Canadian intellectual network that was sponsored by the Canadian Institute for Advanced Research. And we were interested in both the basic science, but also the policy implications of this belief that prenatal conditions and early life events are critical for promoting developmental health. And we sort of, the title of this anthology, which we published in 1999, was a bit of a play on words from the famous economist, which was the argument that developmental health, if you endorsed it, would promote child well-being, and that was our future, and the future of any society. Conversely, to the extent to which a society does not pay attention to developmental health, it was compromising and putting its future at risk, that is, our children. So we wrote this book, and like most academic anthologies, it's pretty dense. I don't necessarily recommend you go back and find it. But what was interesting at the time, this was 1999, George Bush was running for president, and the head of our group, Clyde, got a call from the Bush campaign, from some of the policy wonks who were part of his staff, saying they had come across the book and they found the message kind of interesting. So they wanted Clyde to go down to Texas, to Crawford, Texas, to tell them a little bit about the take-home message. But when he got there, he was surprised that they said, the book is way too long and too complicated. Can you encapsulate the whole message of your book in a slogan, one sentence? True story, true story. So Clyde, being clever, he said, well, the take-home message of our research in this book was no child left behind. For those of you who are old enough to remember the campaign and the Bush. So it's not easy to get basic science to become policy, and it turned out it became standardized testing and all of that. Nevertheless, I would take that compassionate conservatism in a second today over where we are currently. And the other interesting part to me historically was in Canada, these same researchers had much more influence on policy and the provincial governments than we were ever able to have in the US. And you can even still see to this day a divergence of Canadian policy, and we won't get into single healthcare systems and all of that. But the message we were trying to convey didn't fully come across as we wanted. But that's another layer of this. How do you get basic science to become part of policy? But let me get to the real meat of the presentation. over time, and even if we were to poll this room, there are very different views about pregnancy and what women should do and how we should view this whole prenatal period. And so to sort of push you a little on this, just evaluate this poster. 100 years ago in prenatal clinics in the United States, this poster hung. So this is from 1919, and if you read it, most of us would agree with this part. They were trying to comfort pregnant women, saying, no shock can mark an unborn baby, no horrible sight can deform him. I think most of us would resonate with that message. Babies are buffered and protected. But how about the second clause? But worry, fear, and anger may affect his mother's blood, which supplies his food, and therefore pregnant women should remain calm, happy, and sweet-tempered. I don't know if that message still plays so well 100 years later. Maybe. I would particularly like this part because, as you'll see before the end, I've been spending a lot of time studying nutritional pathways from moms to babies. So this is 1919, about 100 years ago now. To get a bit more current, if you're not familiar with this acronym DOHAD, Developmental Origins of Health and Disease, it sort of came to the forefront with the research of this British epidemiologist. His name was David Barker, and it got to be named after him, the Barker hypothesis. And what David Barker argued were, and this photo is from one of his books, these three kids are not the same, and they won't be the same for the rest of their lives. And he focused in particular on the runts, the preemies, the small for gestational age kids, and he argued that if they had experienced some insult during the fetal period, or for whatever reason they were growth stunted, they would develop what he called the thrifty phenotype. Expecting an adverse postnatal world, they already made some adjustments in their metabolism, in their regulatory set points, which if they were to now come out into a world where resources were available, they would be prone to obesity, prone to hypertension, and to type 2 diabetes as adults. So that was the essence of David's work. He focused mostly on the little kids, and as I said, we focus a lot more on the big babies, because the same ideas, we think, hold. So in Wisconsin, there are more overweight individuals than there are maybe in California, and without even having to necessarily pre-select for women who gain a lot of weight during pregnancy, it's fairly easy to acquire a sample of babies who were gestated by women who gained quite a bit of weight. And when I say quite a bit of weight, in our studies, that typically means more than 45 pounds while they're pregnant. So they may start pregnancy a little bit overweight, and then they go on to gain a lot. And this is from one of our papers. This is a fairly recent one from 2016. There were 85 healthy babies, all delivered by C-section, because delivery itself could activate these same pathways. And what you can see was that the women who are relatively larger with a BMI over 35, their babies have more of this pro-inflammatory cytokine in their cord blood. And if you take the cells out of the cord blood and then stimulate them in vitro with stimulants, in this case it was PHA, they tend to produce higher levels of pro-inflammatory cytokines. And we were looking at TNF, IL-1, IL-6. You can see why I asked you earlier, do you know how much your mother gained during her pregnancy? So this was in keeping with this idea that we should add maternal obesity and gestational weight gain to the factors that might increase the risk for atopic conditions, for allergies and asthma. And so in my worldview, maternal obesity as well as other things such as prematurity or gestational length are part of a priming of the baby which will influence who it already is at the moment of birth, and as illustrated here, whether or not it has an atopic phenotype, predisposed to inflammatory conditions. So these would be babies that might develop eczema, rashy babies, wheeze during the first year of life, and then by 3 years of age, it now presents as asthma. But really the story began earlier, and that if you're interested in biomarkers, you can already tell by the types of cytokines. In this case, regulatory cytokines like interferon IL-12 will be low. Other cytokines like the pro-inflammatory cytokines or IL-4 and IL-5 will tend to be higher. And then the world is superimposed upon that. So if you happen to live in California now where you're exposed to a lot of smoke, that's superimposed upon this predisposition. If you don't believe me, just a few examples from the literature. This was a epidemiology study done in New Zealand looking at the likelihood of developing allergies by 5 to 7 years of age, and it's just plotted against the child's birth weight. We tend to think of the small babies, the David Barker idea, the small babies, but actually in this study they found it was the large babies. If you're not used to thinking about grams, this is 9 and 10 pound babies are actually at higher risks for allergies. So are preemies. In fact, it's biphasic. So if you look at gestational length now as opposed to weight, both preemies and babies that go long are more at risk to developing atopic conditions, allergies, and asthma. Segue now to using monkeys as a model. So for many years now, in fact, it's almost 40 years, I've been using monkeys as a model to investigate the influence of prenatal conditions, pregnancy conditions on fetal and infant outcomes. So this is a short list of some of the different types of conditions we've studied in terms of how it would influence babies. We've looked at infection. I'll show you some of the effects of influenza. We've studied the effects of stress during pregnancy on fetal development, that I will illustrate. We've also looked at antenatal corticosteroid treatments that I won't show you. Instead, I will focus on the importance of iron biology as one pathway in the story. I've already touched on obesity, and if I don't run out of time, I'll talk briefly about our current research looking at the microbiome as the readout of this type of influence. Why study infections during pregnancy? Obviously, infections can be very serious, clinically important. There can be a loss of a pregnancy if it's early on. There can be effects on growth development, growth rates and retardation. It can precipitate a premature delivery, and I know one of the foci here is the possible effects on infant brain development, even if the baby goes all the way to term. So for the older pediatricians, if there are any in the audience, you will be familiar with this acronym called TORCH. These were viruses that were thought to be potentially teratogenic, to have effects on fetal development, including toxoplasmosis, but also herpes viruses, rubella, and now we've all become much more aware of this because of the recent effects of Zika. Since I'm at Davis, I wanted to just highlight Tarantal's work, which some of you may know, about CMV, which probably half of us carry, based on normal prevalence rates. Half of us are CMV carriers, but you definitely do not want CMV to cross the placenta and reach the fetal compartment. This was a study done over 20 years ago where she showed that if you took CMV and actually put it into amniotic fluid of a developing fetal monkey and then came back 2 months later, there were these severe effects on brain development. But even without the virus crossing the placenta, there is the percent, and reaching the amniotic fluid in the fetus, there is the potential for there to be not this severe, but other effects on brain development. And at the time I was focusing on this, we were particularly interested in flu infections. And at the time, and still to this day, it was thought that even though most of us don't view flu as that much of a risk factor, it may increase the likelihood of neurodevelopmental disorders as well as increase the risk for schizophrenia. But we were still in this early period unsure of whether flu was that significant, since flu rarely crosses the placenta to reach the fetal compartment. And so it was possible that the baby was still buffered, protected. If you're not familiar with how common it is for women to get flu during pregnancy, it's fairly common, especially if the pregnancy runs across the winter. In this particular study, they found that 11% of the pregnant women had been exposed to the flu. So if it is of potential concern, it's not a trivial factor, it's a fairly common event. And again, since I'm here, I picked this from some of your research here at Davis, indicating that, it's not an overwhelming factor. I mean, the odds ratios here are not that large, but for families that had an autistic child, there was an increased likelihood of influenza infection, and in particular, if the flu infection went on to be associated with a fever, then that increased the odds ratio of having a kid with neurodevelopmental problems. So this was the model we focused on because, as I said, flu rarely will cross the placenta. It can, but it's rare. Rather, we and most other people were of the opinion that it was the inflammatory response of the mother, in particular certain cytokines such as IL-6. Others have argued that IL-17 is more important, but the idea was that it was the mother's own inflammatory and immune response that was leading to a cascade of cytokine responses, maybe first by the placenta, but including by the fetus. And given that there's no real blood-brain barrier in the fetus, that these cytokines had ready access to the brain. So this was how we set up the study. The nice thing about doing studies in animals is that you can control the infection, you can control the type of flu virus that was the pathogen. And so we had monkeys from controlled pregnancies, we had monkeys that we purposely, experimentally exposed to the flu. And again, the nice thing of an animal model is we could pick a particular time. And so we focused on day 120, when we exposed the mother to influenza. It resolved within a week or two, and then we went on to let them deliver naturally. And at a year of age, we did neuroimaging with MRI to look at the brain development of either the controls or the flu-exposed infants. So just like with a person, you can put a monkey in a scanner, you can image its brain, and then you can look at a number of different parameters. You can look at total brain size, you can look at gray matter, white matter development, you can look at the size of the ventricles, and we did all of that. But what I'll show you is the impact of this 1 week influenza infection on day 120 of gestation on the size of the brain at a year of age. And it's presented as the difference relative to the controls who had not been exposed to flu. Very dramatic, negative impact of a flu infection at this point. In this particular study, because of the sample size, it had to be a change, a decrement of about 6% to reach significance. But as you can see, looking at gray matter and white matter volumes in a number of regions of the cortex, frontal cortex, temporal, in particular parietal, was significantly smaller in the fetal, well, they're now a year of age, but monkeys that had been exposed as fetuses to influenza, bearing in mind that it wasn't the virus that caused, it was rather the mother's inflammatory response which was having this negative effect. So if you were to go to the CDC website right now or after this presentation and look at the recommendations, they would say that women planning to get pregnant now, if you're planning to get pregnant this winter, the recommendation is to be vaccinated. And I used to have on this slide, or during the first trimester, but actually, because that's the way it used to be said, it now says on the CD website, you should be immunized at any point in pregnancy, including all the way into the third trimester. So the belief is that vaccines are safe, that it is preferable to be immunized and protected against flu rather than to risk a flu infection, especially if you're the kind of person who might go on to get a fever. And in part, the belief is that you're not just immunizing mom, but because of the passage of antibody, the mother is passing on protective antibody that will protect the baby for at least 4 months after birth. So the CDC says flu vaccines are safe and a good thing, especially if you're a higher risk woman, which would include somebody who has asthma, because that increases your chance of going on to develop pneumonia after getting flu. So in my own mind, these studies of infection were really a part of a larger context of any type of challenge or insult during pregnancy, which might include stress. So in a sense, in my mind, infection was a part of this larger universe of insults and challenges to pregnancy. And we spent a lot of time looking at the impact on behavioral outcomes in the infant monkeys and on other aspects of brain development. And just to illustrate how we would do this in an animal model, again, the nice thing about working with animals is you can control when things happen. And so we would generate infant monkeys from controlled, undisturbed pregnancies, or we would intersperse a period of disturbance or stress. And in our research, we would pick different time periods of pregnancy, typically for 5 or 6 weeks, when there would be daily stress. We could start it at one point in pregnancy and then stop it at another. And in this case, as you can see, we had either an early period from day 50 to 92 or a late period from day 105 to day 147. And again, they would then be allowed to go to term naturally to raise the infant, and we would evaluate these babies from these 3 types of conditions. And in some ways, this is the more unusual situation, which is to go through a pregnancy with no stress at all. But that's another topic. So we have a variety of ways, some of which are used at the primate center here as well, to evaluate infant outcomes in baby monkeys. So in Wisconsin, we modified what is known as the Brazelton Neonatal Assessment Scale, and we have one for baby monkeys where we do a series of similar tests to look at their reactions. And to just summarize a lot of research in one graph, baby monkeys that come from stress pregnancy are different. They're different at 2 days of age, they're different at 2 weeks of age. This is at 2 weeks of age. If they come from a disturbed pregnancy, they are more emotional, less able to calm down after they've become aroused. In order to test them, we have to take them from the mother so they have an emotional reaction. They're more emotional, less able to calm themselves after they become aroused, and their neuromotor systems are relatively immature as compared to a control baby. And in this research, for this outcome, the late stress was, sometimes it's the early stress which is more impactful, but in this case it was the late stress condition which had a greater effect. Somewhat amazingly to me, given that this was only 6 weeks of pregnancy, we could also see some fairly large effects on brain development as well. This was at 3 years of age, bearing in mind that nothing negative was done from the day of birth until 3 years of age, but still at 3 years of age, we could see that the hippocampus of ones that had come from these disturbed pregnancies was smaller than from the control pregnancies. And in this case, as you can see, looking across the length of the hippocampus, it was really a thinning of the whole hippocampus, which explained this difference in total area. This was a study where we were also able to look at neurogenesis because we had injected the monkeys with BRDU, which is a marker of new neuronal cell growth, and also we found that there was an effect at 3 years of age on the number of new neurons which were appearing in the hippocampus, depending on whether they had come from a control or a disturbed pregnancy. I'm not going to show a lot on this, but I just wanted to illustrate this because this is a little bit more current. We now have some other non-invasive, less invasive ways of evaluating baby monkeys, one of which, I like this mostly just because I thought of it one day, which was that the hair that's on a baby's head at the moment of birth, whether it's a monkey baby or a human baby, was actually exposed to hormones in the final month of pregnancy. So baby hair starts to grow, even in monkeys, in the last month of pregnancy, as is true for us, which means that any hair that's on your baby's head at the moment of birth, like tree rings, tells you about what hormones it was exposed to in the last month of pregnancy. How cool is that? So we went on to show that baby monkeys from primips, first-time moms, have different hormones in their hair, not just cortisol, but different levels of reproductive hormones, testosterone, than do babies from multiple pregnancies. That was kind of interesting, and then we went back and said, oh, well, if that's true, let's take a look and see what happens with a stress pregnancy. And in this particular experiment, they had been disturbed in the mid-gestation, and now we're looking at hair at birth. And so probably you, like me, think, oh, well, that means their hormones are going to be elevated. Cortisol, stress hormones should be higher. That was my prediction. It actually didn't turn out that way. It actually turned out to my surprise, it was actually lower. So the babies at birth had lower cortisol levels. And then, in order to try to think this through, I had to think back to the fact that we had disturbed them in the middle of pregnancy. Now there was a month or two with no disturbance, and what I probably did was shift the set points. I think the baby had downregulated its adrenal activity, hence the hair that was growing in the very last month of pregnancy actually had less hormone rather than more. It still was impacted, but it actually was opposite to my initial prediction. And I think that explanation is credible because we had years and years ago done studies on DEX exposure. We had been giving DEX to pregnant monkeys. And actually what happens then is you get a downregulation of the HPA axis in the fetus because the baby's been sort of overwhelmed with more, in this case, glucocorticoid, but I think in the case of stress, too much maternal cortisol has crossed. That was what we wrote in the paper anyway. Let me segue now to the immune system. So I kind of take that same perspective and these same ideas forward to thinking about the development of the fetal immune system. And if you're not used to thinking about it, the important point to take home is the immune system begins fairly early in fetal development. By mid-gestation, there are lots of T cells, there's a thymus, and the largest the thymus will ever be is at the moment of birth. There's a lot of important immunity that's gotten underway by the time a baby is born. And as you'll hear me talk about it, from my perspective, it's a learning system. So just in case you didn't believe me, you've never really thought about it before, there's a lot of thymus development that happens in utero. And in fact, at the moment of birth, your thymus is relatively bigger than it will ever be for the rest of your life. Babies where our thymuses are relatively small as they're my age. You have to go look for it. But in a baby, it's large. It's hanging over the heart like that. This is human, not monkey. The thymus is very responsive to insults. So this is the equivalent of a bacterial infection during pregnancy, fetal inflammatory response syndrome, all these circles. This is done with ultrasound, just looking. So you can use ultrasound to look at the size of the thymus. And like in you and me, when we're stressed, it tends to get smaller, thymic involution, even during fetal development. And we really don't know the long-term consequences of this and the recovery of this, especially if it was repetitive. This is in sheep, but it's the same idea. It's fairly universal. In this case, they used endotoxin. They injected LPS into the amniotic fluid. And again, very quickly, the thymus is smaller in the fetal sheep, and it lasts, as you can see, for 5 days. The take-home message is the immune system, like the endocrine system and the brain, can respond to insults, and maybe these are programming events. So I spent a lot of time on this. I hate to think of how much of my career I spent on this, but it allows me to provide this summary. We demonstrated that when you disturb a fetal monkey, in this case, typically with just stress, we can show that there were many effects on the developing immune system. When the babies were born, they couldn't distinguish self from other antigen as well. We used to call these CD4 suppressor cells, but now they'd probably be called Tregs. They were different if they came from a disturbed pregnancy. I will show you this. The maturation of natural killer cell activity was different. The cells don't proliferate as well when you put them in culture. Many different immune tests to demonstrate this one point. Insults, challenges during pregnancy affect the developmental trajectory of the immune system. I just wanted this, I'm only going to show this in a cartoon version, just because I thought also this was one of the coolest studies I ever did. And for the young people in the audience, I don't think it's ever been cited. Nevertheless, this is my opportunity to tell you about it. When I said, if the baby came from a disturbed pregnancy, at birth, we looked at its ability to recognize its own cells, because one of the learning aspects of the immune system is you shouldn't react to self, especially T cells. The other learning part during pregnancy is you learn to not respond to your mother cells, because some of your mother cells are also crossing into your own circulation. So when I said to you, baby monkeys from stress pregnancies can't distinguish self from other, the ones that were from these disturbed pregnancy couldn't distinguish their own cells very much from their mother's cells. We also looked at their reaction to their father's cells and to an unrelated animal. They were a muchness, whereas a normal infant monkey at birth, its cells can already tell my own cells, self from other, from unrelated other. I thought that was so cool. Never been cited. It flunked into the literature and died. It was just ahead of its time, again. But I think it's related, I know your group has a lot of interest in autoimmunity and all of that. And I think it's still an interesting way to test that. For women who've had babies, your baby cells circulate in you for about a decade. It's kind of a interesting thing to think about after over a drink, maybe, but not as long as you're not pregnant. This is what I did want to show you because this was the serendipitous finding that got me to study nutrition. So we were looking at many different aspects of immune development in these baby monkeys, and one of them was natural killer cells. These are cells that are primed to kill virally infected cells, cancer cells, hence the name natural killer. And the way you do that is you collect lymphocytes, you incubate them with a target. In this case, this is a Epstein-Barr virus, herpes virus transform target, and the natural killer cell should lyse it. So that's the measure. And so what you can see here is as the control babies are developing, they're getting better and better in this cytolytic activity. Not so much if you came from a disturbed pregnancy. So already by 2 months of age after they're born, you can see there was some significant difference. But this was the one that I didn't understand. But as opposed to just being different, they actually seem to be getting worse. So they were lower if they came from an early stress pregnancy. They had lower NK activity at 2 months of age, but by 6 months it even seemed to get worse as opposed to recovering and maturing. So it suggested that there was a second hit that I didn't know about, the second pathway that was actually affecting the baby after it was born. And that was this, especially for the young and people in the audience, sometimes there's a serendipitous finding that you should pay attention to, and this just bugged me. I didn't understand the 6-month one. And it turned out, and I did not anticipate this, there was a second hit that was happening later after the baby was born, and it was that we inadvertently were also increasing their risk for iron deficiency anemia. And this second hit that was coming at 6 months of age was that the ones that had come from the early stress pregnancy were becoming anemic. And as they became anemic, their NK activity even got worse than it had been before. Fortunately, I had a great postdoc in the lab. Her name was Elizabeth Shortcliffe, who was really good with statistical modeling. And she was able to demonstrate in a causal way what I thought was happening. And this is called hierarchical rate of change analysis, where she demonstrated that to the degree to which the prenatal stress was affecting the red cells, that explained this decrement, subsequent decrement in the NK cells. So the rate of change, the degree to which they were becoming anemic, accounted for why they were declining further. So that changed the trajectory of my own career, and I have spent the last decade trying to understand that. How is it that insults during pregnancy can affect iron biology and the risk for becoming or not iron deficient? There are a number of factors, including stress, diet, illness, inflammation, and obesity, which influence the mother's iron status, which in turn influence how much iron a baby will have at the moment of birth. And this is really important, much more important than I realized, because half of the iron that our own infants and infant monkeys need to develop normally has to be acquired before birth. The other half comes in breast milk, but breast milk does not have a compensatory way of upregulating iron. If the baby is shorted at the moment of birth, it is going to become iron deficient at 6 months of age. So let me re-say that because that's important to everything I say next. 50% of the iron that a baby will need to develop normally for normal growth and brain development has to be there at the moment of birth. Breast milk cannot correct for it. We as clinicians, pediatricians, can give iron, but breast milk cannot correct. It just provides the other half. Here are a list of known risk factors for babies getting shorted, not having enough iron at birth. Teen pregnancies, because teen moms compete with the baby. Premature births, because this iron comes in the last month of pregnancy. If your baby was born 4 weeks early, it's shorted. Multiple births, because the iron split. Diabetic pregnancies, we've done a lot of work on that because not as much iron gets to the baby, and what gets there gets utilized, as well as gestational hypertension and stress and obesity. So that's what we've been studying in Wisconsin, now back in humans, not in animals, which is the influence of obesity, large weight gains, gestational diabetes on this iron biology process. And this is just one of the studies showing that women who were we called at high risk, which includes large weight gain, were much more likely to have babies born with low serum ferritin. This is in cord blood. The iron stores are low. Ferritin is the storage form, and these babies are at high risk for going on to becoming iron deficient. We also look at measures such as the cord blood zinc protoporphyrin. When iron is low, zinc goes up, and we can use that as a biomarker that tells us, and I can tell you in baby monkeys or baby humans, if they have elevated zinc protoporphyrin, that baby's going to go on to become iron deficient at 6 months or 12 months of age. It took me a decade, again for the younger people in the audience, you have to be patient sometimes. A decade ago, we showed that stress in pregnant monkeys could affect iron transfer to babies. It took me a decade and a great graduate student to demonstrate that that finding translates to humans as well. And so what this study, which we just published, shows is that if women tell you they experienced a lot of stress during pregnancy, we showed that it affects the iron measures in their cord blood. If they went on to breastfeed for 6 months or more, then when their kids were a year of age, they had low storage levels of ferritin. So I need to just walk you through that one point. Interestingly enough, because all formulas are so heavily fortified now with iron, it's one of the few times where it might be okay or better to have formula, where since in a sense that is compensating for the low iron at birth, but for women who nurse through 6 months, especially women who then don't give solids, foods, at 6. You know, our grandmothers used to introduce solids by 6 months of age, but a lot of people now think it's okay to nurse exclusively to 12 months. If you experienced a lot of stress and your baby had low iron, you're going to end up looking like this at a year of age. So this was the well baby visit at 12 months of age and the likelihood that they would be iron deficient. Just quickly to say, it's more than just iron, and it does matter. We have a whole panel of measures we look at in cord blood. This is EPO, erythropoietin. So the baby's body is responding. This is EPO made from the kidney to tell the baby to make red cells. These are just babies born from pregnancies where the women were large and gained a lot of weight. And you can see from the scatter plots, whether we looked at pre-pregnancy BMI or the delivery BMI, that it was affecting EPO levels, which then would tell you something about oxygen supply. It's not just EPO, we measure hepcidin. I mean, these are all published papers if you're interested. So what can you do about this? Because at my age, I'm at a point where I'm trying to do something, not just show that there are all these bad things. One of the interventions that we've looked at is spoken of as delayed cord clamping. If there are any midwives in the room, you would just say, well, that's natural delivery. So we looked at delayed cord clamping as an intervention. So, you know, ever since the early 60s, the first thing that happens when your kid is born is they clamp the cord. That was how my kids, I didn't think about it at the time, that was how my kids were born. They clamped the cord instantly because they were concerned about the extra blood the baby would get. But that blood actually serves a very important purpose because Mother Nature recycles the iron from those red cells. And so one protective approach, intervention for intervening in this process is simply to provide the baby with more red cells. It's actually a fairly big transfusion. So if you're not familiar with this, the placenta passes about 30 minutes later. It pulses for a while, and as much as 50 to 100 mils of blood will be passed during that process. But if you clamp, it doesn't get there. And again, for those of you who work with monkeys, just think about what the monkey mom does after the baby's born and the placenta is passed. She lifts the placenta over the baby's head, but she's going to eat it and compress it. And think about the transfusion of blood that the baby monkey is getting. And then macrophages come along and scavenge the red cells, extract the iron, and return it to the body. We also are looking at novel types of iron supplements in our research now. It's not such a great idea to dump iron into the gut because it can stimulate gut bacteria as well as be beneficial to the host. And so we're looking at novel ways of supplementing babies. We're studying whether we can use the storage form of iron ferritin as a supplement itself and thereby prevent the bacteria from benefiting from the iron that we give. That's my segue to this last thing, which I'll do fairly quickly. Like lots of people, I became interested in gut bacteria and the microbiome, and everything I've said about programming and the influence of prenatal conditions, the mother on infant outcomes, you can just say ditto to bacteria. So you know, we've become aware that there are billions of bacteria on us and in us, and they're very important for health, and that's especially true during early development. This is a pictorial summary of the literature that we now have available. Pretty much every type of prenatal or early life event that you can think of has some effect or been shown to have some effect on the gut bacteria of babies. If they're born premature, different bacteria than if they're born term. If they go into the NICU, then they'll pick up the bacteria from the NICU as opposed to the parents. If a mother decides to breastfeed, that provides prebiotics and affects bacteria. Formula-fed babies, different. This is what these articles look like if you're not used to looking at them. This is a PCOA plot. It's just showing that babies in different colors, different spheres, whether they're born by C-section or vaginal natural delivery, they end up with a different mixture of bacteria, types and abundance, the microbiome. And some argue that these effects can last for up to 1 and 2 years. We haven't seen the effects go out quite that long, but in baby monkeys, we can definitely demonstrate that if they're on the mother breastfed, they're very different than if they're born by C-section. and go into a nursery. So that's what we study now, the microbiome of monkeys, of baby monkeys. This is just a pictorial representation to show you, in this case at the genus level, that by 2 weeks of age we can distinguish the bacteria of a normally raised monkey that was naturally delivered from one that was C-section delivered and went into an incubator. There are some bacteria that are stimulated by breast milk, bifidobacteria. For those of you who take probiotics, you know a lot of them have bifidobacteria in them, or lactobacilli. Those are ones that are stimulated by breast milk. And what we also see is that the ones that are with the mother more quickly acquire the natural species repertoire of bacteria than do the ones that are nursery-bred. I'm not the only one who's become interested in this. There are researchers here, some of you may know them, at Davis, who've done similar studies. I was showing it to you at only 2 weeks of age, but we see differences that lasted 4 weeks of age, 8 weeks of age, and I believe this study was done here at Davis. They went out and showed that these bacterial differences linger at 6 months of age. Even after the transition to solid foods, you can still see differences. And in this particular paper, they showed that the differences in the gut bacteria were related to the development of immunity or immune tolerance, Treg cells around the gut, as well as some aspects of metabolism. They focused mostly on arachidonic acid and prostaglandins. Same type of message. Early life events, the mother influencing a pathway then that goes on to have lingering effects beyond. And we're first coming to appreciate the significance of gut bacteria for all aspects of the host metabolism, as well as what is spoken of as the gut-brain axis. When I'm not in the lab doing research, I'm an undergrad teacher. So this is just to end with the take-home messages that could be on your final exam. So the messages were, if you're not used to the term, fetal programming, that events that happen prenatally can program in a way that has consequence later. This is related to plasticity and the fact that not just the brain, but a number of other physiological systems, especially the immune system, are really learning systems. They have to be primed. That's what they should do, and that could either be beneficial, or if it's the wrong kind of priming, it can be deficits, detrimental. Why do these effects last? How can they still occur at 3 years of age, 3 years after the original event? The idea is that it's because it shifted regulatory set points, and it's not just the systems I spoke about. There are others who do similar types of research on the kidneys. It turns out the number of nephrons we have, the size of the nephrons are programmed prenatally, and that the kind of kidney function I may have at my age could have been predetermined all the way back by what my mom was doing. Developmental trajectories, and I guess the last term that I have used in some of my own writing is the idea of biological expectancy. Mother Nature, evolution, expects these kinds of influences to be there. It expects the mother to provide bacteria to the infant. And so if you were to deliver an infant monkey by C-section and stick it in a nursery, you have violated that expectancy of this maternal-infant relationship. Oh yeah, I guess I threw this in at the last minute last night because it comes back to the issue of policy and societal issues and clinical practice. If this is true, if this prenatal period is so important, it then means we need to have health systems in place which provide prenatal care. I mean, there's no way a woman's going to know that she should take prenatal vitamin supplements. She should be immunized. She should consider her weight gain and exercise unless she goes to the OB or her primary care physician early in pregnancy, first trimester. So in general, you could say this in the positive. America's pretty decent. About 77% of women go to the doctor in the first trimester, but there's a 22% that don't get seen by a doctor until the third trimester. And it's not an equal opportunity thing. Who are the women who don't get seen until late in pregnancy? It's not a great surprise. It's, if you don't have insurance, health insurance, you're less likely to go to a doctor. And it's not equal across the whole country. It depends on the economic. California's actually looking pretty good. These are the likelihood that a woman will be seen in the first trimester. But if you go to the South and other parts of the country, not so good. So if you believe that prenatal factors matter, that this really is related to developmental health and the wealth of and future of our country, we need to sort of translate this into policy. So I'll stop there. Thanks.
Just wondering, in terms of the policy issues, it sounds like the extrapolation is that additive levels of stress will cause perhaps more challenges for the child. Like, I can imagine there's the stress of going to a horror movie while you're pregnant versus the stress of not having somewhere to sleep at home, and the addition of that. Have there been studies to look at those additive factors?
Not in quite the way you phrased it in terms of titering, that there are enough different studies that we know the types of events. So there have been studies, for example, on communities after an earthquake in California. There have been studies of the women who lost husbands when the World Trade Center went down. So we know on the extreme level, so if we talk about loss of a care provider, those types of events are enough. But the way you asked it is, what is the minimal amount? I don't think we actually know enough. In an animal study, you could titer it that way. I chose to do 6 weeks of daily stress. I mean, there was some thought behind that. I wanted it to be recurrent. I didn't think 1 day of stress would be enough. I didn't tell you it, but I also purposely selected to do the stress in the late afternoon. My own belief is the body is much more tolerant of stress in the morning, whereas we're supposed to reach a period of quiescence and recovery in the evening. So I think even time of day of when the stress would happen and how repetitive it was. I do know in animal models, 6 weeks, which is 25% of their 24-week pregnancy, is enough. But I would say that's still an open question as to just at what point does it become tip over. And it would also, I should also say, it would depend on your outcome measure as well. I mean, I, again, titered the stress in the animal studies because I didn't want to lose any pregnancies, and it was purposeful that we didn't do it in the last month of pregnancy because we didn't want any to be premature births.
So I was just wondering if you could give some comments on the potential effects of fetal intervention, for example, open fetal surgery or in utero transplantation. Those kind of situations, what could happen? Or
well, clearly those examples that you gave would be clinically needed, and before we worry about differences in developmental trajectories, you need to be alive, and if you're talking about open heart surgery in order to survive, I mean, that would have to take precedence over the potential consequences, unintended consequences. So I would, if it were my own child, I would say first you need to breathe. When I was studying antenatal corticosteroids, for example, and it was pretty clear that if you give Dex or other steroids that it would have effects on the baby, but on the other hand, if a baby doesn't breathe, which is why they were giving Dex to preemies that was certainly a far worse outcome than worrying about the negative consequences of a DEX exposure. So I guess I'm sort of talking around because I don't have really a specific answer to your question. But if it were my own child, obviously I would take the open heart surgery and then work on trying to address whatever side consequences of having a smaller thymus. I'm sure the thymus would be smaller after open heart surgery, but that would be of secondary importance to just being alive.
Thank you for the comprehensive discussion about all of these different exposures in pregnancy that can create problems. Could you speak a little bit more toward what your thoughts are about what predisposes to resiliency? Why some fetuses will be exposed to sometimes dreadful things, but perhaps they're saved?
So I would say that, to echo the way you expressed that, and in some ways it builds on the comment I made to the prior question. Obviously, the most important thing is to have a viable baby that goes to term. And in none of these manipulations that I spoke about did I talk about babies that were aborted, didn't make it to term. So that already is a success story as compared to earlier time periods. Infant mortality used to be very high, 100, 200 years ago, and in some parts of the world, Afghanistan, other places, it still is. So in a sense, that speaks already to your comment about resilience. I mean, in general, women are the stronger sex. You are. In general, babies are pretty resilient, that they even make it to term, despite some of these insults, being exposed to endotoxin, other things. So I agree with you. I would say, as a policy level answer, the worst case scenario is a bad in utero environment followed by a bad postnatal. Sort of a two-hit, difficult in utero conditions followed by poverty, followed by poor health care. So I would say that's probably the worst case scenario, is where it's extended. I don't know if you know the name Megan Gunner. She studies adopted kids from orphanages from around the world who've been adopted into the United States. And so I've been working with her looking at their immune responses as teenagers after they've grown up in middle-class homes in Minnesota. And we did find some effects, but in general, and we're writing up that we found some effects, but really, I should say, we should have written the paper about the effects we did not find in these adopted kids. So the kids who had grown up, started their life in orphanages, 80% of them had been exposed to CMV, probably while in the orphanage. So 80% of them were seropositive for CMV, and we were able to demonstrate that was not a good thing to be CMV positive as an infant, and we could show that it affected a number of T cell subsets. But in general, these teenage former institutionalized, now adopted, looked great. So a part of our message in that paper should be there's a lot of opportunity for recovery, which is built on this idea that you're talking about, which is the resilience factor, some of which is genetic, some of which is good parenting, some of which is having supportive teachers, supportive society. So yeah, that's probably actually a good point to actually end on, which is the idea that there's resilience and lots of opportunity for intervention and recovery.
Excellent. In regards to the same exposures that you've talked about, you've mentioned all the, I guess you would call them deficits in a child. Do you have any information relating to positives that could result from the same exposures, where stress could actually create a characteristic in a child that is actually beneficial, as opposed to something that you talked about in terms of negative stuff.
So in some ways, that's similar to the first question, which is this a U-shaped curve? And do you need a certain amount of stimulation to develop normally? And the answer is yes. My postdoc mentor was a fellow named Seymour Levine, who did a lot of research. At the time, it was called the early handling effects, the gentle handling effect. If you're not familiar with that, he studied mice and rat pups that were sometimes put on a shaker and stimulated by vibration, mechanical stimulation. And he showed very clearly that as a certain amount of stimulation was actually critical, the absence of which was bad as well. And if you're familiar with the work of people like Michael Meany with grooming behavior, licking behavior in mice and rats, there too is you need a certain amount of stimulation, the absence of stimulation, which in a sense is the absence of bacteria growing up in an incubator if you're a baby monkey in a nursery. That too has negative effects. So it probably is one of those U-shaped functions that a certain amount, well, in this case this way, a certain amount is required, is needed. That's priming, if you will, of the system. But at another level, it crosses over to become too much, detrimental. But I don't know, for each of these systems that we've talked about, I don't know if we actually can sort of say exactly where on the scale that happens.
All right. If there aren't any other questions, I'll take the last one. Chris, I'd like to hear your thoughts on gestational timing. So we tend to think of the first and second trimesters as being more vulnerable, but your influenza model and a lot of your prenatal stress research would suggest that that continues well out into the third trimester. So I'm curious to hear what your thoughts are.
Providing, with the caveat that it's speculation, armchair. In general, I think most people would say early is worst. In general, most people think that the earlier in the process the insult happens, the larger the impact, and it sort of becomes magnified over time. And that's certainly true for some of our studies where we found early disturbances were worse. But as you commented, we also had done some studies where it seemed like later had more consequence, and it could speak to aspects of pregnancy, but more likely if it's a behavior outcome, it's stages of brain development. So there are obviously critical periods of brain development, such as when the oligodendrocytes first appear, which is sort of around day 105, day 110. So if your measure was myelin, you might find actually it was the late insult that had a bigger effect on myelin. So to dance around the answer, because I really don't have a definitive one, I would say it probably depends on your outcome measure as to whether you would say that early is worse than late. But I would totally agree with you that gestational timing is definitely something one should consider, just as we even considered time of day. I actually found that, as I said, stress in the afternoon had a bigger impact than stress in the morning. We did similar studies. We injected monkeys with ACTH, so it was just a hormone challenge. And the same dose of ACTH had a much bigger effect on the baby if we did it in the afternoon and evening than if we did the same dose in the morning. So even within a day, there was a sort of a chronobiology type of effect.
The UC Davis Mind Institute was created in 1998 with a promise to find cures for neurodevelopmental disorders. Every day, our physicians and researchers come closer to fulfilling that promise. Their groundbreaking research on autism, Fragile X syndrome, chromosome 22q11.2 deletion syndrome, ADHD, and other brain disorders are helping children achieve their fullest potential. Please visit our website to find out more about current studies, upcoming events, and how you can help make a difference.